Okay, so let's go through these notes. Yeah, we're gonna look at. We're not even gonna get into meiosis today. We're just gonna go over mitosis. So it's kind of like a Science 9 review, but it's a little more complicated. So, so just a quick little cartoon thing there. Okay, so cell growth and mitosis is what we're going to look at first. So, cells grow until they reach their limit size, okay? They either stop growing or they divide. Cells can only get so big. What tends to happen is the bigger the cell gets, think about diffusion, right? The further things have to diffuse inside the cell. So actually the cell just it gets to a point where getting too big is a problem. Yeah. Yeah. We look at kind of diversity of kingdoms, so taxonomy, and then evolution. No, that's where we get to do a little bit of like dissecting a few things, and like worms. We'll do a little bit of that, and frogs. And they're like this big. They're not like a bullfrog, but they're not a little frog. I don't think that would work with ours because they've been in formaldehyde for a while now. Yeah. So, no, we're not going to kill anything. Um, so we'll look at a few things like that. We just talk about different things and stuff like that. And then it ties nicely into evolution. Okay. So what happens is the ratio of the surface area, so remember surface area is the outside covering of the cell, to volume. So the volume of the cell would be the actual cytoplasm itself and the contents in the cell. This limits the size of the cell, okay? So as the volume increases, so what happens is, gentlemen, as the cell gets bigger, the volume of the cell actually grows way quicker than the surface area. So there's less area for things to diffuse through, but it has to travel further. Or we could travel through more to get there. So it's a ratio between surface area and volume. And the bigger the cell gets, it has more volume but less surface area. Typically not, no. Okay. No. A nucleus will get a slightly bigger right before cell division, when the chromosomes become more defined. But other than that, like they'll dissolve and then, yeah. So what ends up happening is, so if the cell becomes too large, it has trouble maintaining homeostasis. Okay. We don't have that problem because we have a transport system that helps to move things. We don't rely on diffusion, so it's a little bit different for us. Okay. Now, the cell cycle prevents cells from becoming too large. It's the basis of life for every organism. This is the way the cells reproduce. Okay. They enable organisms to grow. Replaces worn out or damaged cells. Allows healing and regeneration, which is important in our bodies. Takes a different amount of time in different types of cells. Okay. So, for example, neurons last the life of a person throughout the cell cycle. So, therefore, the cell cycle is very long. Skin cells and intestinal cells are replaced very quickly. Therefore, the cell cycle is quite quickly there. Okay. Other great ways to measure like cell growth in your body, how quickly your hair grows, your nails grow, all that sort of stuff. Okay. That's basically epithelial cells as well. All right. Yeah, yeah, exactly how quickly a scar or a scab heals or a scar fades, right? They say like what, 70% of dust in a house is just dead skin cells or something like that. Yeah. Yeah. Well, you're constantly just shedding. So, yeah. All right. So, now let's quickly just talk about our friends, the prokaryotic cells. Remember, they're very simple. The DNA is not confined to a nucleus. Okay. It divides usually by a method called binary fission. Okay. It's asexual reproduction. So one cell splits into two after the DNA divides, creating two identical cells. It happens very quickly, and it can depend on a few environmental conditions, okay? Okay, prokaryotic cells can also reproduce through conjugation, okay? So this is a sexual method of reproduction. So they actually can have a partner here, and you can exchange some genetic information. Now, two prokaryotes, what they do is they actually join together and they exchange genetic information. Am I missing something? Yeah. We were just talking about that today. That's right. Yeah. So, hey-oh. Look at you guys. Okay. Okay. So they actually exchange genetic information. So two prokaryotic cells, they'll kind of like make this little juncture where they join, and they exchange information with what's called plasmids. All right. So what they do is they kind of just pinch off a little bit of their genetic information. It becomes a little plasmid, and it travels from one cell to the other. Okay. Okay. Now, the genetic information transferred is often beneficial to the recipient. Example, antibiotic resistance. All right. So not only can that cell that survived the wave of antibiotic treatment reproduce and pass that on, but it can also find other cells and pass that on to it as well. Okay. So it's a very, very simple and successful way for prokaryotics to thrive and survive. Now, unfortunately, it's not so simple in eukaryotic cells. We go through this cell cycle, okay? So it involves three main stages. There's interphase, and when we did this in grade 9, we just talked about interphase, it's just growing. But it's actually quite complicated, okay? So interphase is 90% of a cell's life cycle. It's where the cell grows, it carries out cellular functions, and it replicates DNA. Okay? Then the other two steps, one is mitosis, so that's where the nucleus and nuclear material divide. And then there's cytokinesis. This is when the cytoplasm actually divides and the cell splits. Okay? So here you can say in interphase, though, there's actually 3 phases within that itself. Just like with mitosis, there was PMAT, right? In interphase, there's actually 3 phases, the G1, the S, and the G2. The G1 and 2 phases are just growth, okay? And the S phase is growth and DNA replication or synthesis, so S phase. Okay. Now, interphase. This is kind of like the childhood and kind of teen or puberty years of a cell, okay? So, what happens in interphase is the cell grows, it matures and develops. It's actually going to duplicate its DNA, and it prepares for division. All right, made up of the 3 phases, G1, S, and G2. So in G1, normal growth functioning, preparing for division. In the S phase, the DNA actually copies. And then in the G2 phase, prep for division. And this is where actually they'll develop what's called spindle fibers. Do you guys remember those from grade 9? I just said they'd show up. These spindle fibers show up and they pull the chromosomes apart. Yeah. Yeah, yeah, yes, yes. Centrioles, very good. Okay, so these develop in the S2 phase. Okay, so this is the nucleus here of the cell. Um, the very center is known as the nucleolus. The center in here, all of this around it, is the chromatin. It's just genetic material. All right. So before mitosis, you'll have a chromosome, and then after replication, you're going to have it in pairs. Okay. Double chromosome. Now, mitosis takes place. Okay, the reproductive years. So here are the spindle fibers and everything start to kind of migrate. So the first part of mitosis is prophase. Okay. So the membrane will start to degenerate. All right. Chromosomes become thicker and visible. They're actually condensed. You can actually see them now. The nucleolus, the very center, will disappear. And the spindle fibers reach out between the poles. So by the poles, like they'll go to the north and the south, the top and the bottom, or east and west. They'll kind of go to one opposite side of each other, and that's where like they'll pull apart. Okay. So this is what happens to like the chromosome, the genetic material. It's all like loose, and then it's really becomes thicker and more dense. Okay, and quite visible. And this is what it actually looks like during or under a microscope. This would be an onion root. So this would be late prophase. So you can really see the chromosomes there. You see like all these dots and it looks kind of faded. That's earlier prophase. The nucleus is starting to degenerate and you can start to see the formation of the chromosomes, those fingers, okay? So far are we okay? Levi? Yeah. Yes. Yeah. Here we go. Back on. Second phase of mitosis is the metaphase. The middle. Meta, right? M&M, right? Mm-hmm. So, the chromosomes, remember, there's pairs of them now, line up along what's called the equatorial plate. All right? These spindle fibers actually help that happen now. These spindle fibers will grow and they attach to the chromosomes and they actually push them kind of into the middle plate there. All right, and you can see it here as well. You'll see quite a condensing in the middle, and you can actually see the little fingers there on the outside pushing it. Okay, pretty cool. Now, anaphase. Okay, what happens now? Apart. Okay, paired chromosomes are pulled apart by the spindle fibers. So the spindle fibers, they grow and then they start to shorten. All right, that's what's happening there. Chromosomes will travel to opposite ends or the opposite poles. When this starts to happen, the cell actually starts to elongate a little bit. Okay, you can actually see it quite nicely on that blue cell over there. That you can see metaphase where they're all in the middle, then you can see how they're starting to pull apart. See how it goes from being nice and round to kind of looking more like a football? It's kind of the fourth one in. Yeah, right? More almost could say like a rugby ball, I guess. Now, what also starts to happen here is cytokinesis starts here as well. Okay? So like the last phase of the cell cycle actually still starts during mitosis. Okay? No, I was, yeah, where was I? A banana phase. What did you see? That's anaphase, yeah. I bounced around. Okay, we're just in anaphase and now I'm going to telophase. Okay, sorry, I hit the screen. Okay, so that's last screen. Okay, so in telophase, the chromosomes reach the opposite poles. Okay, now the cleavage furrow begins to separate the two new cells. Okay, so that cleavage furrow is like a dent, kind of starting to pinch off the new cells. Okay, the chromosomes begin to unwind, so they won't be as pronounced. The nuclear membrane begins to form again, and the nucleolus reappears. Okay, so the whole nucleus starts to come back. All right. Uh, Typically, you can see that one under a slide as well. You actually see like the two halves being pulled apart on opposite ends, and you'll even see if it's an animal cell, you'll see that cleavage furrow. Okay, if it's a plant cell, whenever plants have a plate or a cell plate in them, or a cell wall, you'll actually see a plate kind of start to form a very pronounced line in between the two of them. And then it'll just grow all the way up and split the cell. Okay, instead of pinching it, this plate kind of divides it. Okay, now, so let's get into cytokinesis. Oh, here we go. Okay, so just quick, this isn't on yours. Which stage of mitosis do we believe this one is? Sammy Adriana first. I would go with anaphase. Yeah. For this one here, see how the fingers are being pulled apart? I'd say anaphase for being pulled apart. Okay. So this is a plant cell here. While in telophase, you'll also see, though, you'll see the cleavage furrows. Yeah. Okay. Okay. Let's try another one here. Okay. Now this one's a little trickier. It's just one singular blob of genetic material there. There's no pulling apart. There's nothing going on, really. Cytokinesis is the splitting. Interphase, yeah. Where the cell spends 90% of its time. Okay, so yeah, that's just interphase. Okay, so cytokinesis, this is the cytoplasm being divided. Now, in plant cells, I just talked about this. A cell plate gradually forms across the original cell at the equatorial plate. So in the middle part there where they actually pulled the chromosomes apart, that middle line will actually just start to form a plate that will pinch the cell off. Cell walls will form on either side of the plate, which will eventually create two identical cells. Okay, in animal cells, indentations or cleavage furrows begin at each side and pinch the cell into two. So kind of, here's your pinch, and there's the plate that will form there. That will form the wall. Okay. And then this is also another cleavage furrow there under probably an electron microscope. So far, so good? Okay, good. All right, so these are the stages of mitosis. Here is just a little review. Okay. Okay. So G1, S, G2, and then mitosis takes place afterwards. Now, what's really neat is cells have checkpoints between all of these to kind of make sure that they're ready for all of this to happen. This is just a little side thing. So like before like synthesis and the duplication of the genetic material, there has to be a cell signaling that G1 is finished and all that. That's why like, remember how I talked about how neurons in your body, they don't divide. They never leave G1. They grow and then there's no signaling that goes on for them to enter these phases. Okay. Well, they don't They don't grow. They're created just like any other cell is originally. Like there'll be one, and then it'll divide and divide. But once you're developed, it's done. Okay. Yeah. Just like any other cell that will become specialized. What's interesting though is once these checks kind of start to break down over time with age or the introduction of a carcinogenic, is where you get cancers, right? You get this rapid cell growth that's uncontrolled. There's no checks and balances, and that's typically where cancers will come from. There's some of them. I shouldn't say all of them because there's a wide variety of different types out there and causes for them, but that's kind of where a lot of cancer research is going right now, is to at these genes and cell signalings and seeing if they can control it. Okay. So are we okay with the whole PMAT thing, cytokinesis, S1, or sorry, G1, S, and G2? Okay. So then this is just another onion cell. It shows the different phases. So like this is prophase, metaphase in the middle, anaphase, they're being pulled apart. Here's cytokinesis. You can tell this one's cytokinesis by how close both nucleuses, by how close both nuclei are. Okay, like these ones are a little more spread out. This here is also probably anaphase. Yeah. So right here is where the cell plate will start to develop. Okay. Yeah. I'm just trying to see if I can see one where you can actually see the plate. No, it doesn't look like it. Because sometimes what you'll actually see is like, see here how the two nucleuses are being pulled apart there? You'll actually start to see the line form right in there. Okay. So, any questions about this slide? Okay. This is like, they do this sometimes in bio classes or in university or sometimes in bio 30 as well. They'd actually have you count how many different like phases of mitosis you see under the slide. So you go through and be like, hey, where's all my interphase? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You count them all. And then like you do percentages and stuff like that. Because what you should end up getting if you were to count all the interphase that was in here, it should be 90% of what you're seeing, roughly, give or take. Yeah, they should add up to 10, roughly. And like, so you do that for an hour and your eyes is killing you because you're staring down a microscope, right? Trying to count. No, depending on your field of view, you wouldn't have to do the whole slide. I've seen it too where they just have flashcards printed off with them and you just count them like this, which is much easier than looking under a microscope. Okay, that's all I kind of have for today. Is how everyone feeling about that?